The final two senses are gustation or taste and olfaction, smell. Both of these are chemical senses. They have chemoreceptors that respond to chemicals in an aqueous solution. Taste responds to substances dissolved in saliva. Smell to substances dissolved in fluids of the nasal membrane. They are complementary to one another, but respond to different classes of chemicals. They both give us information about the chemical composition of our surroundings, and they help us link the external environment with internal needs such as hunger, thirst, and even danger. So let's start with gustation or taste. So the receptor for taste is a taste bud. Each taste bud consists of between 500 and 100 epithelial cells, which can be one of two main types: gustatory cells, which are the taste, or basal cells. There is actually a third kind of cell called a supporting cell that helps to insulate the receptor. Each gustatory cell has a very long microvilli called a gustatory hair that projects out through a taste pore to the surface of the epithelium where it is covered in saliva. These hairs act as the sensitive part. They function in two ways. One, they will admit ions that give rise to the sensation of salty and sour, or they bind the molecules that give rise to the sensations of sweet, bitter, and umami. Around each of the gustatory cells are sensory dendrites, which are obviously the beginning part of the journey to the brain. As you can imagine, the taste buds are constantly abused, and we replace them every 7 to 10 days. The basal cells are what replaces our taste buds. So they are new gustatory cells waiting to differentiate. We actually have a number of different types of taste buds. So the function of taste is really designed to be a protective mechanism, but it was designed such that we could test food and other substances before they entered the body. Substances that are useful for the body tend to taste good, and substances that taste bad are potentially harmful. We have about 10,000 taste buds, and most are on our tongue. We do have some on our epiglottis and some in our lower pharynx. Specifically, taste buds are found in the papillae or on papillae. These are small projections on the tongue that give that bumpy or slightly abrasive feel. Taste buds are mainly located on the tops of the fungiform papillae or mushroom shape. These are distributed across the entire tongue and have between 1 to 5 taste buds associated with them. We also have something called folate papillae, which are on the side of the tongue. We have circumvallate papillae, which are large, round papillae at the back of the tongue that form a V shape. And lastly, we have filiform papillae. These are conical papillae and are the most numerous. Each consists of a long, slender core of connective tissue covered by epithelium. They are keratinized on their tip, and they have no taste buds associated with them. They are more of a protective mechanism. So earlier I said that those gustatory hair cells responded to different tastes. So there are 5 basic taste sensations. Sweet, so many different substances, including sugar, alcohol, and some amino acids provide a sweet taste. Salty includes metal ions or inorganic salt, such as table salt. Sour is an acid. In particular, we utilize anything with hydrogen ions. We can taste bitter, which are alkaloids such as nicotine, quinine, caffeine, morphine, and aspirin, although that is a non-alkaloid. And the last one is umami, which means delicious, and it's elicited by amino acid glutamate. So if you've ever had MSG, people often used to add something called MSG, monosodium glutamate, to food to give it a better taste. But some people found that they had a reaction to it, or it made them feel not good. Umami is also what gives beef taste to steak. So the differences are not absolute. Some taste buds have the ability to identify 2 or more sensations, and obviously some substances may also produce a mixture of sensations. Originally, it was thought that there was a greater predominance of one type of taste bud in a certain location. However, this doesn't seem to be the case. There is only very slight variations in some sites, and it's likely that all types of taste can be elicited from all over the tongue, although there may be greater concentration in some areas. The bitter location is, however, important, as many foods that have gone off or are rancid have an alkaloid taste. And having those taste buds predominantly so far back, you are more likely to initiate the gag reflex. That means that those foods will not be swallowed and can be spat out. There are, of course, going to be individual differences from person to person. So in order for something to be tasted, or a chemical to be tasted, it must be, first of all, dissolved in the saliva and must come into contact with those gustatory hairs. Binding of those food chemicals causes depolarization of that taste cell membrane, releasing a neurotransmitter. This initiates or generates a potential that will ultimately elicit an action potential. Each cell has a receptor on the apical surface. These receptors are transmembrane proteins which are connected to a sensory neuron. Each of the receptors has a slightly different process. For example, the receptors for salt are ion channels that allow sodium to pass through, depolarizing the cell, causing an action potential. Sour ion channels admit hydrogen ions liberated from that sour substance or acid. These prevent potassium from leaving the cell and thus cause depolarization. The remaining 3, umami, bitter, and sweet, all use slightly different versions of G proteins, which have been named gusjuicin. You don't need to know any more detail than this. Cranial nerve 7, or the facial nerve, and 9, glossopharyngeal nerve, carry the impulses from the taste bud to what is called the solitary nucleus of the medulla. The vagus nerve, or cranial nerve 10, carries impulses from the taste buds in the pharynx and the epiglottis. These impulses then travel to the thalamus, and from there the fibers branch to the gustatory cortex to provide the taste, but also to the hypothalamus and the limbic system to give us an appreciation of taste. It's also important to note that this pathway triggers reflexes, so we have synapses with parasympathetic nuclei, because we need to have this involved in the digestion process to include increasing saliva secretions and gastric juices. Interestingly, taste is about 80% smell, or in other words, flavour is a combination of taste and smell. We also utilise thermoreceptors, mechanoreceptors, and nociceptors to influence taste. Temperature and texture can either enhance or detract from taste. There are also a couple of related medical conditions, such as ageusia, which is a loss of taste function of the tongue, hypogeusia, which is partial loss of taste, or dysgeusia, which is distortion or alteration of taste. Sometimes some medications can cause this. First and foremost, smell is protective. It provides us with a distinct chemical sense. Humans can tell the difference between about 10,000 different odours, whereas dogs are about 10,000 times more sensitive to odours than humans. As we've just stated, smell complements the sense of taste, and qualities of food that we think are taste are actually a function of smell. The olfactory mucosa is an organ made up of olfactory epithelia, which is a pseudostratified epithelia, and the lamina propria. The lamina propria contains numerous glands that secrete mucus. The mucus protects the olfactory epithelia and also provides a solution which allows odours to be dissolved. These odours must be dissolved so that they can be detected by the olfactory receptor neurons. These are what we refer to as first order neurons, and we will return to them in a little bit. In mammals, the olfactory mucosa is located on the roof of the nasal cavity, above and behind the nostrils. It's not actually in a very good location, as the air has to make a turn to actually pass over the receptors. When we sniff, we actually change the direction of air, making it easier to smell. So the actual organ of smell is the olfactory epithelium. The olfactory receptor cells are bipolar neurons with radiating olfactory cilia, which will aid in increasing the surface area. Olfactory receptors are surrounded and cushioned by supporting cells. These supporting cells provide physical support, nourishment, and electrical insulation for the olfactory receptors. They can also help to detoxify any chemicals that come in contact with the olfactory epithelium. Basal cells lie at the base of the epithelia and continually undergo cellular division to produce new olfactory receptors, similar to what we saw with the gustatory cells. These receptor cells are unique as they have a fairly rapid turnover with a typical lifespan of 30 to 60 days. The cilia, or olfactory cilia, tend to lie flat and are covered in mucus. It is this mucus, as we've said, that acts as the solvent that collects and dissolves the odorants. The axon of these cells become the filamentous component of the olfactory nerve, or cranial nerve 1. These axons pass through holes in the cribriform plate of the ethmoid bone, and they join or synapse with a structure called the olfactory bulb. So, similar to other special senses that we've talked about, it is the olfactory receptors that are activated from gas molecules that pass through the nose during respiration that ultimately will result in electrical activity, which is then sent to the brain. Activated olfactory receptors are this initial player in a signal transduction cascade, which will ultimately produce that nerve impulse. The olfactory nerve, or cranial nerve 1, is the first of 12 cranial nerves. And the olfactory nerve consists of a collection of many sensory nerve fibers that have extended from that olfactory epithelium to the olfactory bulb. As we said previously, they pass through the many openings of the cribriform plate of the ethmoid bone. The olfactory nerve is the shortest of the 12 cranial nerves and only one of 2 cranial nerves. The other is the optic nerve that do not join the brainstem. The olfactory bulb is supported and protected by the cribriform plate and separates it from the olfactory epithelium. The olfactory bulb transmits smell information from the nose to the brain and is thus necessary for the proper sense of smell. For us to be able to smell something, the odorant must be volatile, i.e. in a gaseous state. It then has to be dissolved in the mucus of the olfactory epithelium. The olfactory receptors respond to several different odor-causing chemicals. Odorants are absorbed into the mucus and diffused to the olfactory cilia, where they initiate this transduction process by acting on membrane receptors. Activation of the receptor leads to G protein mediated stimulation of adenylate cyclase, producing cyclic AMP. This then activates a gated channel, allowing ions such as sodium and calcium to diffuse down their electrochemical gradient. So if we're looking at this picture, step 1, the odorant binds to the receptor site, causes cyclic AMP to be produced from a G protein. The sodium channels open, depolarization occurs, and an action potential will result in the axon of the receptor. Calcium is a little bit different because it allows the transduction mechanism to adapt, decreasing its response to a sustained stimulus. This means that when calcium comes in, we become oblivious to a degree of the smell. So if you work in a stinky environment, you still can enjoy your lunch because we have adapted to this environment due to the calcium coming down its gradient. Olfactory receptor cells synapse with mitral cells. These are second-order neurons. However, we do have an initial processing step before this at the glomerulus. Glomerular cells process the odor signals. Notice that each glomerulus or little ball receives axons from neurons that have come from the same type of receptor, indicated here by the same color. Each glomerulus receives only one type of odor signal. Different odors will excite a number of receptor types and therefore a different subset or group of glomeruli will be excited. After this small amount of processing and combination, the mitral cells will send this impulse via the olfactory bulb to the olfactory tract. From here, we go to the olfactory cortex. This is the location where smells are consciously interpreted and identified. As with taste, we also send the information to the hypothalamus, the amygdala, and the limbic system. This will elicit the emotional responses to odors. Some, such as smoke or skunk odor, can trigger sympathetic fight or flight responses. We could also have a protective reflex, such as sneezing, be elicited. Other smells that are more appealing, such as certain food odors, will trigger salivation and stimulation of the digestive tract. So, in review, we have now looked at the location and structures of both the gustatory and the olfactory system. We've looked at the different tastes and how they might be recognized. We've looked at the activation of receptors in both systems and the pathway to the brain for both systems. Again, how these are interpreted are beyond the scope of this unit.